Every living organism, from tiny bacteria to humans, contains a genetic instruction manual. This instruction manual is called DNA. DNA controls how organisms grow, develop, function, and reproduce. Inside your body are about 37 trillion cells, and almost every cell contains DNA. If the DNA in one human cell were stretched out, it would be about 2 meters long, yet it fits inside a tiny nucleus. DNA has a unique structure called a double helix, which looks like a twisted ladder. The DNA molecule is made up of repeating units called nucleotides. Each nucleotide has 3 components: a sugar molecule deoxyribose, a phosphate group, a nitrogenous base. There are 4 nitrogen bases in DNA: adenine, thymine T, cytosine C, guanine G. These bases pair in a specific way: A pairs with T, C pairs with G. This pairing forms the rungs of the DNA ladder. DNA is found inside the nucleus of eukaryotic cells. Because DNA is extremely long, it must be tightly packed. DNA wraps around proteins called histones, forming structures known as chromosomes. Humans have 23 pairs of chromosomes, giving a total of 46 chromosomes. Before a cell divides, it must copy its DNA. This process is called DNA replication. DNA replication occurs during the S phase of the cell cycle. Steps of DNA replication. Step 1. Unzipping. An enzyme called helicase breaks the hydrogen bonds between bases. This causes the DNA double helix to unwind and separate. Step 2. Base pairing. Free nucleotides in the nucleus pair with the exposed bases on each strand. Step 3. DNA polymerase action. An enzyme called DNA polymerase joins the nucleotides together to form new strands. At the end of replication, 2 identical DNA molecules are formed. Each molecule contains 1 original strand and 1 new strand. This is called semi-conservative replication. Another important molecule in genetics is RNA. RNA is similar to DNA but has some differences. DNA is double-stranded while RNA is single-stranded. DNA contains deoxyribose sugar while RNA contains ribose sugar, and DNA uses thymine while RNA uses uracil. There are 3 main types of RNA. 1. Messenger RNA. RNA carries genetic information from DNA to the ribosome. 2. Transfer RNA. tRNA brings amino acids to the ribosome. 3. Ribosomal RNA. RNA forms the structure of ribosomes. The first step in protein synthesis is transcription. During transcription, DNA unwinds. 1 DNA strand acts as a template. RNA polymerase builds a complementary mRNA strand. Base pairing during transcription is a TU, T to A, C to G, G to C. The mRNA then leaves the nucleus and travels to the ribosome. The second step in protein synthesis is translation. This occurs in the ribosome. Steps: mRNA attaches to the ribosome. The ribosome reads the mRNA 3 bases at a time. These groups are called codons. tRNA brings the correct amino acids. Amino acids join together to form a protein. Proteins control nearly every activity in the body. DNA is important because it stores genetic information, controls protein production, allows inheritance of traits, and ensures cells function properly. Without DNA, life as we know it would not exist. DNA is one of the most important molecules in biology. It stores the instructions of life, replicates itself before cell division, and works with RNA to produce proteins. From DNA replication to transcription and translation, these processes ensure that every living organism can grow, survive, and reproduce. Understanding DNA helps scientists study genetics, diseases, evolution, and biotechnology.